Hey guys, so today we're going to be learning about carbohydrates, which is a subunit that we have to learn about in the IB biology curriculum. What are carbohydrates? Essentially, carbohydrates are just sugars. They can be simple sugars like monosaccharides, or they can be a chain of sugars like polysaccharides. Um, they're just sugars essentially. About monosaccharides, there's a lot of different types of sugars, um, a lot of different types of simple sugars, sorry. You've got things like pentose and hexose, which the difference between them is that hexose has one more carbon within the molecule than pentose does. And within those, like pentose, you've got fructose, you've got ribose sugars, and in hexose, you've got things like galactose, and more importantly for us, glucose sugars. But essentially, there's just a lot of different types of sugars. Now, the one we need to learn is glucose, and for some unholy reason, we have to memorize how to draw it. Now, there's not really a specific way to memorize the glucose, like sugar, but I like to do it this way. I like to set it up into 3 steps. I first draw the carbon ring, which has an oxygen, and always draw the oxygen on the top right. So always draw it here because it's just going to be much easier for you to remember. Always keep it in the same location. Then I draw the outside. So I memorize all of the outside first, and then I move on to the inside. It won't work for everyone, but it works for me because it helps me break down and memorize each step instead of having to go straight into memorizing everything all at once. It's just an easier way, I suppose. There are different ways of memorizing it though. My teacher, the way he learned it, or the way he prefers to know this, even though he doesn't have to know it anymore, is by just memorizing where these hydrogens are. And he does it by going up, up, down, up, because this one's above the hydroxyl group, this one's above the hydroxyl group, this one's below the hydroxyl group, and this one's above the hydroxyl group. That's how there you go, have 2 different ways of memorizing it. But I guess it's just something that you have to know. We also need to know the properties of glucose, but these are pretty easy. It's soluble, it's charged, it's very stable, and it can't be oxidized. Interesting thing about it being charged, if you know about the cell membrane and all that, is that it cannot pass through the cell membrane through simple diffusion. It needs to be passed through facilitated diffusion with a channel protein. So that's a callback to the cells topic. But how do you get polysaccharides from a monosaccharide? You get that through the condensation reaction, which is a reaction that builds things up. And it does so by creating these bonds, which are called glycosidic bonds. And it does this, you might see that there's just an oxygen bonded, but clearly there's 2 hydroxyl groups here. That doesn't make any sense. It's because in a condensation reaction, you actually produce H2O or water. So in a condensation reaction from a monosaccharide to a polysaccharide, one of these hydroxyl groups, let's just take this one, and one of the hydrogens from the other hydroxyl group, which would be this one, break off the glucose in our case, and it would form water. And then this bond over here would bond, or this carbon bond over here would bond with this oxygen, and then these hydrogens are obviously left alone there to create the glycosidic bond. But from this we can get disaccharides, which are just 2 monosaccharides put together, no more, no less. Or you can get polysaccharides. which are more than 2. So it's like many, you know, poly is many and saccharides are just sugars, so many sugars. We don't really need to memorize the different types of disaccharides, but as I said before, because there's many monosaccharides, there are a lot of different types of disaccharides and polysaccharides. So if you've got 2 fructose bonded together, you're going to get a different disaccharide than if you've got 2 alpha glucoses bonded together. In a polysaccharide, however, we do need to know 3 of them, which are starch, cellulose, and glycogen. The good thing about these 3 is that they're all glucoses. So we don't really need to know what type of sugars they are. But we do need to know their structure and also their function. So let's look at that. So first we've got cellulose, and I know that there's 4 right now, but I will explain that later. So first we've got cellulose. Cellulose is found in plants, and it's a structural polysaccharide. So it's indigestible, it has a lot of hydrogen bonds, and these each individual, so this is one individual monosaccharide of glucose, can rotate 180 degrees this way. So it can, I don't really know how to draw it. So not horizontally, but vertically. But yeah, so the structure of it, I know I said that we don't really need to memorize the type of sugar per se, but it is good to know what subtype of glucose it is, but there's only 2, so it's okay. So cellulose is beta glucose. I'm just gonna write the shortened version because I'm lazy. And it's linear and unbranched. So it's just a straight line of glucose and there's no branching off of it. So it's just a thick line of polysaccharides. And it's got 1 to 4 linkages. What I mean by linkages is the links between, or what carbons create the glycosidic bonds. So if we count, you always count clockwise when you're talking about this. But this would be a glucose, so this would be 1, 2, 3, 4, and 5 glucoses. This is the first glucose, and this would be the this is the first carbon, and this would be the 4th carbon, which is why it's called a 1 to 4 linkage, because the bond is between the 1st and the 4th carbon. So I hope that makes sense. But that's cellulose. Then you've got starch, and starch can be found as amylose or amylopectin. So let's start with amylose. Or I guess both are found in plants and they are both used as energy storage in plants. So amylose is less soluble, it takes up less space, but it is harder to digest for plants. It is an alpha glucose. You've got, it's linear, but it is helical. So there's no branches, but it also doesn't have a straight line formation, as you can see here. And just like cellulose, it's got 1 to 4 linkages. That was a weird 4. Now in comparison, amylopectin is more soluble, it takes up less space, and is easier to digest. So this makes it the more preferred, it makes it the more preferred version of starch for plants regularly. So this amylopectin is also alpha glucose. It's branched and also helical. And in comparison, it's got 1 to 4 linkages, but it also has 1 to 6 linkages, which is how it gets that branched look to it. The 1 to 6 linkages, it comes from the 6th carbon. So if we look back over here, this would be the 6th carbon. So I guess here in this diagram, the 6th carbon, is just in the middle of the 5th and the oxygen. So this would be the 1st carbon, and this over here would be the 6th carbon. Then we've got glycogen. Glycogen is not found in plants, but it is found in animals and is used for storage. So glycogen is similar to starch. The only difference is that glycogen is found in animals. It is insoluble and it is formed in the liver and the muscles. Glycogen is also alpha glucose. It is branched and also helical. And it has 1 to 4 and also 1 to 6 linkages. I would say this one, glycogen, in terms of structure, is very similar to amylopectin. The only thing is, as you can see in the diagram, glycogen is more branched than amylopectin is. But, yep. So you've got different polysaccharides there. However, there is also more functions that carbohydrates can have, and those are with glycoproteins. Now, if you know about the cell membrane, you would know that the glycoprotein is a cell receptor, and it's got a protein over here, obviously that's a protein, and it's got a polysaccharide, or what they call an oligosaccharide, which is, I suppose, a type of polysaccharide, except it's very few. From what I read, it was like 3 to 10 monosaccharides bonded together. So whereas polysaccharide could be like hundreds, this is only just like 10, maybe 20. But these are important for cell recognition, especially in the blood. So in this case, the antigen would be the glycoprotein, and these are really important when, for example, donating blood. These have antibodies, and if you give the wrong blood to the wrong person, those antibodies could start attacking the red blood cell because of the antigens and the glycoproteins on that red blood cell. So if I gave type B blood to a type A blood person, the antibodies that it produces, which is called anti-A, would be attacking the A antigens in the red blood cells, and vice versa. If I gave the person with if I gave a person who has B type blood, A type blood, then the antibodies B would be attacking the B antigens. That's why the O type is the universal donor, because it doesn't have any antigens. So if you give type O blood to A or B, the antibodies would be attacking essentially nothing. And that's why the AB plus is the universal recipient, because it has both A and B antigens, and so it can kind of survive, so to say, the anti-B and anti-A, because it has both. So you could give it A or B blood, and obviously you can give it O blood because O blood has anti-A and anti-B type. But yeah, those are carbohydrates. Not that complicated if you look into it. They're just sugars. A lot to memorize, I do have to say, but I hope this video helped you, and I will be putting these notes. I will be sharing the whiteboard of these notes down below. Thank you for watching.